so I'm just going to tell you a little bit about how we use LabKey to help us with our work in immunotherapy trials at Seattle Children's. So I'm going to give you a little bit of science to start with. So we're at the little brown building down the road that way. We've just launched, a new department in the hospital called the Immunotherapy Integration Hub. And basically the idea of this department is that we have all the translational pieces to really look at immunotherapy research as a whole. So starting down at the R&D process development, we have a team of scientists that are trying to look at the next new thing that's going to come out, like how can we make immunotherapy trials better, safer on the research side. We have a preclinical team that does, once they have something that they think is worth pushing towards a trial, they do all the safety testing that would be required to file for a clinical trial with the FDA. The Clinical Coordinating Center is a group of people that are there to write clinical protocols and informed consent forms and all the regulatory paperwork that goes with running a trial. Once all those pieces are together, we can actually start trials and run them, and the Coordinating Center also organizes all the management of the trials that are run mostly at Seattle Children's, but we do have a few other sites in the US. And then finally, the last piece is our piece, which is the Correlative Studies Lab. So we receive samples from all the patients, and with those samples, we try and understand what is it that worked in this trial, why did these kids do well and these kids not, so that we can feed it back into the research and hopefully help that cycle keep going so that we can speed up improvements in getting trials up faster. So looking at the timeline of our program, so I actually started at Children's in 2013. The first week I was there, they handed me the first sample from the first patient, which was very exciting. And then, we had a few other trials open and we were kind of stable for a little bit. And then you can see towards here, we started churning out a lot of trials. We currently have 8 trials open. 5 of them are leukemia-based trials. 2 of them are solid tumor trials. And the brainchild one is also solid tumor, but specifically brain tumors, where we're actually doing local delivery of the product rather than just systemic. So just looking at the timeline from this, you can see that everything's kind of had to accelerate a little bit and expanded in the scope. And I'll show you how LabKey's helped with that. So just very briefly what we mean, what we do with immunotherapy. So we take immune cells from a patient that has cancer. These cells are taken and then genetically modified so that they're able to recognize the cancer cells. And then we grow them in the lab and activate them in order to get enough cells to give back to the patient. And then we give them back to the patient, and hopefully the cells that we've given them can now recognize the cancer and kill the cancer. And it's a successful therapy. That's the goal. So in a nutshell, that's kind of what the therapy is. And we then, because we've modified these cells, we're able to track them in the patient. So we can then look in the patient and see where they are, what they're doing, how long they last for, that kind of thing. So in our group, the things that we're most interested in looking at is looking at those cells that we take from the patient to make our product. Because this is a personalized medicine, every patient's going to have a different starting material, and there's a lot of things that can affect those cells. So a lot of our patients have had transplants previously. They've had a lot of different chemo regimens. These are phase 1 trials, so patients have usually had a lot of different treatments before they come to us. So all those things can affect the quality of the product, and if we can determine what affects the quality, then we can maybe find a better time point to get these cells or find a way to rescue them despite the fact that they've been damaged. Then the actual modified cells that we inject into the patient, we know that the way we grow them in the lab has a lot of effect on what the cells look like at the end and how effectively they can then grow in the patient. So we look at basically the characterizations of those cells at that time point and try and correlate with how well the therapy worked. And then I mentioned briefly, in a perfect world, these cells would stick around for a very long time so that the cancer never has a chance of coming back. one big question in the field is how long is long enough? You know, do we need them to stick around forever? Would a year be enough? Is that enough to consider that all the tumor is gone? Like how long do we actually need them to be around? And finally, though this treatment is supposedly less toxic than chemotherapy, there are side effects, and some of them can be pretty severe in some patients. So how can we understand that better and try and mitigate those toxicities so that it's safer for everyone? Okay, so now to LabKey. One of the things with these trials is that you're collecting a lot of data from a lot of places. And to make the most powerful analysis, you kind of need to look at the whole package and not just one set. So once a study participant enrolls, we have 2 parts. In the clinical, all the clinical trial data ends up in an ECRF system. Mostly it's in Medidata Rave. Most of our trials in Medidata Rave. And then on the other side, we have the research samples, so the samples that come to us, and we're generating data with them, and we have a bunch of data on that. So what LabKey is doing is pulling those 2 things together into one place so that we can really analyze the full picture. There is one part that's not represented here, and that's the cell production cause data. They have been traditionally paper-based. They're working on having an electronic system where we could also bring that data in as well. And at the bottom here, it's just to give you an idea of all the data points we collect. So this is a timeline for one patient. We collect data for up to 5 years from each patient. And the purple is us, so we're collecting data at every time point. This is on the green, there's clinical data being collected at every time point, and then the blue over there is that manufacturing team that are collecting during the time that they're manufacturing this product. And then those are all scheduled samples, and around the time where there's the peak activation of the cells, we often get some unscheduled visits where we're trying to find out more at that particular time where there's a particular symptom and that kind of thing. So as you can see, there is a lot of data collected for each patient. We're now at 200 patients, so there's a lot of pieces we're trying to put together and work out. So our server, our LabKey server has grown a lot in the last 5 years. I'm not going to go through all of this, but this was just to give you an idea of the different things that we do outside of just putting data in and using it. So on the top in the orange, we actually use surveys to track our samples. And I'll get into a little bit how we do that, but it's all survey-based, and it's a way that we know what samples came in and what assays we ran on those samples. Then in the red, we have the raw assay data, which is more the traditional. You have assay data, you put it in, and you push it to a study. We also have 2 other sets of data. This is something new that we're going to be actually receiving data from a clinical lab, and we're trying to create a workflow for that data to come in a similar way that we do our own data. And then we also have the ECRF data that is coming in from those databases. So that's all the data coming in. On the left-hand side, we've got some tracking and some QC stuff that I'll show you a little bit of, just to have a way of programmatically checking that there's not anything weird going on, mostly duplicated samples and stuff like that. And then down here in blue is the study folder. So that's really where we're putting everything together and we're sharing data with people, mostly for the actual clinical studies. But we also have our manuscript folders, which are collaboration folders, but we will also pull data and we can then share it with collaborators, et cetera, that are working with us to try and make sense of the data. Okay. Okay, so now I'm just going to go through different parts of our server to just let you know how we're using it. Please feel free to interrupt if you have any specific questions or anything. But so our landing page just has a little bit of information on the different trials we have. You can't actually tell on here, but some of these are links to clinicaltrials.gov or to our PI research page and stuff like that. And then just a few stats on what we do, how many specimens we're receiving, that kind of thing. So as I mentioned, the specimen receipt is all done through surveys. So we designed a survey to track samples as they come in. This survey is basically just tracking, so our participant ID and visit and specimen type all go into the specimen ID. So the first three are all drop-downs to avoid any typing errors and stuff, and then the specimen ID is actually a combination of those three that's automatically generated in the background. Then we have some information about where it was drawn, et cetera, et cetera. Oops, wrong way, sorry. And then at the bottom here, we also specify what are the assays that we're actually running on this sample. So this then logs that sample into our system to say, yes, this sample arrived, and these are the things we want to do with it. The last part of the survey that didn't fit on the page, we do have who submitted a survey, so we can always go back to that person if anything went wrong. And then all our surveys are also verified by someone. So someone actually goes and checks, especially the specimen ID and the date of draw with the source documents to make sure that that is accurate information. So once that happens, we have surveys for our other standard assays. So this is our FICAL and our flow. And again, first of all, you can see that now the specimen ID is a drop-down. Once it's been registered in the specimen registry, it automatically is available here. So that, again, prevents errors between people typing it in. And then again, you can see that these surveys have been are verified as well to make sure that the information is accurate. I just wanted to mention briefly that one of the things we spent a little bit of time on was what do we do when a sample has to be recalled. We have samples that are drawn and then the patient wasn't eligible or they had to do a redraw and we end up with two samples with the same ID. So we have actually designed a system to be able to recall that sample. So in that specimen registry, we have the sample status, which is normally blank, but you can recall it here. Once you recall it here, automatically you can see this do not modify. This is because this is the area where it would then say recalled once you change the specimen registry. So that's one of the ways we make sure we're not duplicating samples in the system. Okay, and then as I mentioned, we do have programmatic checks to make sure that things aren't going awry. Mostly on the top, we're looking at multiple entries in our different registries. You will see that we do have a few, but most of them are recalled samples. So you can see these two are the same one, but one of them's normal and one of them's recalled. So that's something we expect, but we can check that to see if there's any unexpected ones in there. And then from the study report, we can look if any sample has any missing date. So you can see a few of them. This is a study that doesn't have an ECRF, so there's more manual work done there, which is why that one has a few patients with missing information. Then we can go back and add that in. We also track deviations in our system. So whenever we deviate from a protocol or an SOP, we put in a deviation and we mark them based on the risk category, based pretty much on data. High risk means we lost data. Medium risk means we nearly lost data, but we were able to recover it. And so this is, we have our deviation surveys up there that I won't show you, but we also have a way of quickly looking at how we're doing with deviations. So we started measuring deviations over here, which is why there are so few. And then it kind of went up as we got used to putting in deviations for different things. And we actually found that this correlated with sample intake. So we had a very busy summer, and there was a lot of things going on, and we also lost a member of staff, so we were very short-staffed at that time. Therefore, a lot of mistakes happened. We managed to normalize things a little bit with extra staff, and so that's kind of gone down a little bit. But it's nice that at a glance we can kind of see, is something going wrong, are things getting better, getting worse, etc. And so with that graph, we actually thought I don't know how many of you use visual boards to kind of track your progress and see how things are going, but we had a board like an actual physical board. Our team is split into several locations, so we met there once a week, but really most people didn't see the board outside of when we met at it. So we're like, well, can we do an electronic board? So we tried to see if we could do that in LabKey, considering a lot of the data we were tracking was in LabKey. So this board was designed by Wenjun from our visual board. And so you can see on the left there, that's the number of samples we're getting on a monthly basis and on a weekly basis in the past 14 days. And then here is looking, one of the things we're tracking is how long our documents are open for revision, because we have a lot of documents open at any given time. And so these graphs were made somewhere else because our tracking system is in another software. But we just entered the graphs in here. And in the bottom part, more tables of things we're tracking. So all this was coded in HTML, I think, and to look like our visual board. Now, one thing that scares a lot of people, we have a team of 11. We only have one person that knows how to code in any language. So a lot of people are like, well, this is great, but if I want to change something, how am I going to do that? Because LabKey is scary. I don't know how to change things because I don't know how to code. So one thing is for these drop-in graphs, so any image that's there, we built it so that you can just drop the file with the same name and it will automatically change things. So anyone that's updating those graphs can just drop in the file. As long as they're naming it the same way, it will replace the previous one. And then for those a bit more adventurous, you can search through the code for a keyword. And most people can kind of like from that find what they're looking for and just change that one word, Just, okay, there's 100%, I can change it to 80% without having to understand all the code that's in there. So this is very new. We'll see how much people do play with the code. But we're excited to try and see how this works. Okay, so now coming to the assay data. Flow is something that we've spoken about before here. Flow is a lot of what we do. So we spend a lot of time in the flow module. Basically, we acquire data on one machine and one software called FACSDiva. We then analyze data in another software called FlowJo. The great thing about LabKey and the reason we started using LabKey is that they can actually read flow files and FlowJo files and take in that data. So we then drop it into the flow module, and then we actually link it through ETL with our specimen registry, so we can link a specific file to that specimen information. And then there's some quality control involved, and then we can push it to steady. So in terms of quality control, if anyone does want to do flow in LabKey, we have had a bit of experience with it. FlowJo likes to change their versions on a regular basis, and not all of them work as nicely. So it's always good to check that it works before you upgrade to the new version. And then we've also had issues on our side of wrongly named files and that kind of stuff. Once you push the study, it becomes harder to take it back. So we make sure we're looking at everything before it goes there to make sure that the data that goes in is correct. You do have the option of looking at thumbnails if you actually, the graph's down here, so you can actually look at what the graph looks like within LabKey and not have to go back to the raw plots to make sure that everything's going well. And then again, in the flow staining survey, the person that reviews the survey is also reviewing the data, is also reviewing the worksheet, so we make sure that all that matches up and is accurate. Okay. So one of the big changes coming up in the lab is all this flow cytometry we're doing on a daily basis is actually going to move to a clinical lab. And so we're trying to think of how can we replicate our system of getting flow to the clinicians quickly enough while using LabKey, but with an outside lab. This lab uses a different analysis software, Kaluza, that is not compatible with the flow module. And also this lab is not actively using LabKey, so we want to make it very simple for them to just give us the data without too much extra steps. So they will be generating for us a standardized Excel report with all the data that we request from this experiment, as well as a PDF report with all the images of the FCS files. And they will just be dropping that into a file drop location, so we'll just open a folder for them, and that's where they'll drop all the files. So once the files are dropped, the File Watcher module will pick up the Excel spreadsheet, and process it into a list. We do have the capacity to take that data and put it straight into the study. But again, because we want to make sure that the data is correct, we're actually going to do a manual QC step first to make sure that the data matches what we expect, and then push it into the study. And so this will be an automated ETL processed once a day. So we'll know that all the data is being pushed on a regular basis. The one part that is not possible right now, but we'd love for it to be possible, is to also be able to move the PDF file so that when you're in the study folder, you have the raw data, and then you'd have a link to the PDF, and you can actually open it up. That is something that's possible for the admins on the site right now, but for external users, without giving them access to the whole thing, they wouldn't be able to do that. Okay, so another assay we've been working on is our qPCR assay. One of the things that's been a little complicated to scale up is that we currently have a pretty complex workflow for it, in the fact that we receive samples here at Children's, but for several reasons, we actually outsource the DNA extraction to the hutch. So we create an aliquot, we put it on a freezer, and then on a monthly basis, we send a lot of samples to the hutch for them to extract. The Fred hutch actually extracts it, and then we get a box back with all the samples. Once they come in, we check all the samples that are there, try and check that it's the ones that we expect. We put them in our freezer inventory, and then someone is manually adding in an Excel spreadsheet, these are the ones that we should be testing. But there's not a great QC step to check that they've actually entered all the samples, and we don't have a way to check, of all the ones that were sent, are we missing any steps along the way? You know, some samples don't have any DNA because they had such slow cell count and stuff like that, and we don't have a good way of making sure nothing's falling through the cracks. The last step, we run the assay, and the data is stored in a shared drive with a date. So if you're looking for someone's data, you have to know the date it was run in, or you just have to click on every single Excel until you find the one that has the sample that you're looking for. Definitely not practical. It's two big problems. It's very hard to know where a sample is. If someone wanted to expedite a sample, you'd have to try and track where it is in the process. And it's hard to retrieve data. We were working towards putting that data into LabKey, and then thought, well, there's all these other steps. Can we put the whole workflow into LabKey so we can more easily track where it's going? So again, this is new. So we haven't completely tested it out yet, but the idea is we are now, so from that first specimen registry, when we tick a box saying, yes, we're making an aliquot for DNA, we now have a list saying these are all the samples that are in the freezer waiting to go to the hutch. The hutch sends us a spreadsheet normally to say these are the samples we're sending you back. Instead of sending us an email, they'll drop it into a folder in LabKey. Again, we'll use FileWatcher to pick up that file and link it to the DNA sample record to check that we received something back for each of those samples. And then we're actually using the sample sets to assign assays to particular samples. So once you have this table saying these are the samples that are here, these are the ones that were received, you can then use those two buttons there to say, yes, I'm assigning this test for this sample. We can then go ahead and run it, and once it's run, you can say, yes, I completed this test, and someone can come in and say, yes, I verified this test. So everything in one table, you can say, it was here, we received it, it needs to be run, it was verified, all in one place, which is very exciting to us. So I've been given my 5-minute timeline, so I'm going to speed up a little bit. The last thing is the study folders. Most of this is probably more generic, but we have a study folder for each study. In our clinical and assay data, we have all our ECRFs that come in as data sets. So one thing is we've worked now with integrating Medidata, REDCap, and DATSTAT, some more easily than others, but done some modifications around the way. So for example, for Medidata, we now have a UI, so you can actually select which CRF is coming in and which field of that CRF you're actually selecting, which makes it much easier for people to use. We did find that it's not because there is a CRF that it's going to come into a nice table. The design of eCRFs, if they're designed on the data entry point of view, doesn't mean that the data output will be very friendly. So that's something we've been working with the data coordinators with to try and see if we can make it better on both ends. And then data entry, obviously if people aren't entering the data in a timely fashion, then we're not getting the data either. And there is a lot of QC steps to make sure that things are matching up, that we have the same dates on each side and things like that. We have made custom reports to give the data that the clinicians are most interested in one place. I won't get into too much about that. And then we also have, for our clinical sites, we can actually tailor their sites so that they can only see the data from the patients that they enrolled and not anyone else's data. So CHLA just have one study running where they see just their own patients. At Children's, the second one, they have all the studies we're running, so they have a folder per study, and they only see the patients from Children's. And then finally, the manuscript studies. I won't talk much about this, but this is kind of the play area for people wanting to do analysis. So it's a study folder. It means you can pull data from all kinds of different places. And this is where we use R more to actually visualize things and see what's going on. So with that, what we've learnt, a lot of things start with design, and if you don't have design in mind from the get-go, you're going to make your life very hard on the other end. Quality control is essential, and we spend a lot of time on this to make sure that the data is usable. The out of the box does not mean ready to use. We've had, some modules are harder than others to use. We've only just recently had one person fully dedicated to managing LabKey, which is why we've made a lot of improvements recently. But so that you get what you put in. I think the more time and resources we're putting into LabKey, the more we can build it in a way that's really useful to us. There's lots of things moving forward to use. We do want to try out the sample set for our assay pipelines. Finding out a way to move those PDFs over. We're still playing with the SDI board and seeing how it's going to work with everyone in general. And then for our study pages, most of our clinical PIs are very busy people that want to get to their data fast and find what they're looking for. So we're trying to find a way to make it more friendly for them to use so they can find stuff more easily. And with that, a lot of people to thank, obviously, on the CSL side. So Hannah, as mentioned, is the one that did most of this work before she joined LabKey because she loved it so much. And since then, Wenjun Huang has been doing a great job at keeping up her work and putting further improvements. And then on the LabKey side, everyone's been really great with helping us through all our interesting findings and weird situations. So thank you all for that. And with that, I'll take any questions, if there's any time left.